Jackie was talking about an EEG-based biomarker, and Anna is part of the project to see if there's any potential to use blood biomarkers in NDDs to better understand progression and state of the condition. So Anna, would you like to introduce yourself a little bit more? And
Sure. Let me see first if I can share my screen.
yep, go ahead and share your slides.
Can you see my first slide?
Yes, I can.
Ah, beautiful. That was easy. That was too easy. Something else is going to get messed up. Let me see also remote.
Can you do like a presenter mode out of there?
Yeah, I will.
Okay. Yeah. Oh, there
And
we go.
then
Yeah.
I want to see, is there a way to do like laser pointer thingies, or should I just use my mouse? Can you see my
I
mouse?
can see your mouse. Yeah.
Okay, I'll do that.
Yeah.
Okay, cool. Yeah. So I'm Anna Pfalzer. I'm the Chief Scientific Officer for Combined Brain. I'm also an assistant professor in the neurology department at Vanderbilt University Medical Center. And what I am going to show you today is our initial findings from a cross-disorder plasma proteomics project that included almost 30 other neurodevelopmental disorders, including Kleefstra syndrome. And the really extra cool thing about this is actually Dr. Harris helped us collect some of these samples that were included in this project. So that's really cool to present after her. Yes, I think I will. Let's see if I can figure out how to advance my slides now. I'm going to go here. Okay. So I show this slide quite literally every time I give a talk about biomarkers, because the identification of biomarkers, which Dr. Harris already introduced, I'll talk about a little bit later, is an essential component to the drug development process, especially for rare genetic neurodevelopmental disorders such as Kleefstra syndrome. So I won't go into every single step along this schematic here, but you will see on the left-hand side, you see the identification of the causal gene that's causing the syndromic behaviors, the development of rodent and cell models, natural history studies, other cellular pathogenic mechanisms identified, the identification of biomarkers, patient-centered outcome measures, so outcome measures that are clinically meaningful to patients and their communities, and then clinical trials on the right-hand side. So Dr. Harris had almost essentially this exact slide, so I won't belabor it, but I think the word biomarker is thrown around a lot, especially in rare disease communities, and I always have to sort of remind myself, what is a biomarker? It is something more than just blood and guts and other random fluids that you can collect from an affected individual. It is an objective measure which can be used, as Dr. Harris said, to do a number of things. It can be used to predict disease status. It can be used to see if an individual is progressing or not. And then it can also be used to predict an individual's response to a particular treatment. These are common examples of biomarkers that are typically studied in biofluids. So Dr. Harris was talking about other types of biomarkers. There's also physiological biomarkers, et cetera. But for the purposes of this talk, I'll just focus on biofluid-based biomarkers. And these are some common examples of where we find biomarkers. So mRNAs are genes, microRNAs, epigenetic changes. So for example, changes in methylation pattern within a particular population, different proteins, metabolites, and then even microbial populations, so microbiota. So the different types of biologic or biofluids that you can find these biomarkers in is obviously cerebrospinal fluid. That is considered one of the most informative samples that you can collect from individuals with neurological disorders, although you can imagine that that is considerably more difficult to collect than blood samples from patients. Or urine, saliva, or stool. So one of the questions that is always brought up when we're looking at blood-based biomarkers is really, can blood recapitulate what's happening in the brain? And we do see examples of this. There's several examples of this in other neurological disorders. So here I'm including the example of plasma neurofilament levels in individuals with Huntington's disease. So not only do you guys don't have to pay attention to the color on this scatter plot here, but you can see on the y-axis is concentration of this protein neurofilament on the x-axis in CSF, and then on the, sorry, that's a y-axis, and then on the x-axis is you see NFL concentration in blood. And you can see that they correlate very nicely, a significant positive correlation. And moreover, is that the plasma levels for this particular protein also correlate with another indicator, a clinical indicator of disease progression. So even though plasma isn't directly derived from the brain, we do see a handful of proteins and potential for things in the plasma to be biomarkers for molecular happenings in the brain. So one of the limitations for looking to see if there are actually blood-based or plasma-based proteomic biomarkers in rare disease populations such as Kleefstra is obviously access to samples from patients in a rare disease population. So in order to start looking at whether there were plasma proteins that could be potential biomarkers for some of the neurological disorders that were part of our consortium, Combined Brain, we set up this general pilot biomarker design where we tried to collect 20 patient samples from as many neurodevelopmental disorders that were part of our consortium. So we tried to collect 20 from no exclusion on age or sex or variant type, et cetera, just 20 individuals from each disorder interested in participating. And then, of course, we also tried to collect neurotypical controls, so typically siblings or otherwise age and sex matched unaffected individuals. So then we collected a plasma sample from these individuals, and we ran a small portion of that plasma on the SomaLogic proteomics platform. This particular platform, one of its benefits is that it can measure approximately 10,000 different analytes or proteins within a particular sample. In addition to the plasma sample that we collected from individuals, we also had caregivers complete the Vineland, the online caregiver reported Vineland. And then for individuals who reported epilepsy, we had them complete Dr. Greenspan's PILS seizure survey. And then really the primary research questions for this project as a pilot project was, do we see differences in plasma proteome between control patients and affected patients? All affected patients lumped together and not disorder specific. The flip side is, do we see proteins that are unique amongst disorders that differentiate disorders from another disorder? Do we see protein changes with age? Do we see protein changes with epilepsy? And then some other larger sort of network, what are the major biologic pathways being implicated in the plasma proteome of these neurodevelopmental disorders? So I will say, before I move on, for the initial analysis that we've done on this project, and it's still very much ongoing because it's a fairly large data set, for the work that I'm going to show you today, I was really interested in looking at plasma protein changes that occurred during development. So I limited the age of the participants that are included in any of the work that I'm about to show you in the following slides to individuals between 0 and 17 years of age. So truly just a pediatric population. So the way that we collected these samples, primarily the vast majority of these samples were collected throughout 2023, was by flying a research team from Combined Brain to 19 different family conferences throughout the US. So if there is a state with a color here, there was a rare disease conference occurring, and we flew a team and collected samples as well as phenotypic data at their conference. Also, for individuals who wanted to participate but were too severely impaired to be able to travel or otherwise needed to stay in their home, we contracted with mobile phlebotomists who collected these samples and data within the patient's homes. And then, as I mentioned at the very beginning, we were able to collect a handful of these samples in collaboration with clinical labs or other research studies at a handful of academic medical centers, including Dr. Harris at Kennedy Krieger. Okay, so as I said, this large table right here really only represents those individuals that fall between 0 to 17 years of age. And I've highlighted our Kleefstra syndrome population here. I will say, so the sample size for what we looked at in this first analysis is a little bit over 500. And we actually collected a little bit over 700 for the entire study. And Kleefstra syndrome had approximately 20 participants, 11 of which were between the ages of 0 and 17. So the average age was a little bit shy of 10 years old, a fairly even distribution between males and females, approximately 20% reported epilepsy. And then these last 3 columns right here are the composite scores, or the subdomain composite scores for the Vineland. So ABC stands for the aberrant behavior composite, CC is communication composite, and then DLSC is the daily living skills composite. So I'm sure many people listening right now are very familiar with the Vineland, which is, although it is a measure of adaptive behaviors, it is very commonly used as a general indicator for developmental status. So typically in papers, what you see is individuals who have a composite score of less than 70 are considered to be cognitively impaired. So you can see that the average scores for these are somewhere in the 40s. So on average, Kleefstra syndrome population are considerably impaired for this particular study. Okay, one more. Okay. So this more or less shows the same thing from the table below. It shows the composite score over age for the individuals participating in the study, again, by the different subdomain breakdown. This light blue bar right here indicates that threshold of 70 for each subdomain. And then you can see the overall trend line is significantly lower for each of these subdomains for all of the participants across the 28 neurodevelopmental disorders. So amongst the communities that we were working with in this project, it became really clear that one of the priority symptoms that were most burdensome on families were communication skills or lack thereof. So we actually wanted to look a little bit more about what the communication scores and what they looked like within a given community. So we looked at expressive communication by age and receptive communication by age. When you look at these figures right here, this dotted gray line right here is the trajectory you would expect from a neurotypical individual. So the x-axis here is chronological age, and then the y-axis here is the age equivalent score that the individual received for expressive communication, and then likewise for receptive communication. And you can see that this gain in communication skills is significantly blunted compared to the trajectory for neurotypical individuals. However, there is a clear gain in skills across age. One thing that I will actually point out, full transparency, this is not a longitudinal cohort right now. We have collected longitudinal samples, but right now, this is just cross-sectional data. So we actually wanted to look at what this trajectory looked like by disorder. And I know that this is a little confusing, but we actually coded the EHMT1 data as MED13L in this particular case, so I apologize for that. But you can see sort of the orange bar, and you can see how EHMT1 individuals change with their expressive communication and receptive communication with age. And the reason that we started looking at this is because a biomarker not only is just a change in protein between an affected condition compared to control, we're also trying to find what is the appropriate readout that we should use as an indicator for disease severity. And so the reason that we were looking at these changes in expressive receptive communication and the other Vineland scores was to see which readout from the Vineland is the best indicator of disease severity to really interrogate some of the protein changes that we find later. Okay, so now I sort of switch gears a little bit and I start looking at the proteomic changes that we observed in these 500 samples. So the very first thing we wanted to look at was, are there clear changes in the plasma proteome? So all of the proteins that we were looking at on this platform, are they significantly impacted by sex, age, and epilepsy status? So what these little scattered dot figures are right here are UMAPs. These are essentially visual representations of the variability in your data, and each individual circle represents an individual participant in the study. So you can see that there is no clear separation or clustering by red circles, which are females, versus the blue clusters, which are males. The other really interesting thing is that there's also not a clear impact of age. So the white circles are individuals less than 4, and then as you kind of increase in blue tone, you increase in age. So you don't see any really clear pockets of young individuals, adolescents, sort of toddler age individuals. And then the other thing that was a little bit surprising to me is that there's not really a clear impact of epilepsy on the plasma proteome either. So individuals who don't have epilepsy are the white or clear circles here. Individuals that have epilepsy are in the gold circles but haven't had a seizure in the last 2 months. And then the individuals, the red circles here, are individuals that had some type of seizure or epilepsy episode within 30 days of having their plasma sample collected. But again, the same trend holds out. There's no clear clustering by epilepsy status. So the other thing that we were mainly interested in looking at was, are there changes in plasma proteome by disorder? So by disorders that each have a very different genetic cause, do samples cluster by that? Do the Kleefstra samples sort of cluster all in one spot compared to the samples from Dup15 syndrome or GLUT1 syndrome, et cetera? If you look on the right-hand side of the screen here, you can see the different color dots correspond to the different disorders that were in the project. And without looking really closely at which individual colors are Kleefstra syndrome, although it's this cool purple color here, you basically get the impression that there's, again, the same pattern, no clear sort of pockets of disorders separating by genetic etiology. So one of the things that we thought that might cause this is that if you remember, SomaScan platform actually has and includes about 11,000 different proteins. It's actually closer to 10,000. And a lot of these are not expressed in the brain at all. A lot of these, approximately 6,000 of these, are explicitly peripherally expressed proteins. So things that are expressed other places besides the brain. So given that we're looking at the impact of neurodevelopmental disorders, what we looked at next was to see, okay, let's focus on those 4,000 proteins that we know to some extent are expressed in the brain as well as other areas in the body. And you still don't see any clear patterns or similarities amongst genetic cause. And then the last thing that we looked at was, okay, well, still we have other proteins that are being expressed in the peripherally. Maybe that's just making the omics data a little too noisy. So let's just focus on proteins that we know are only expressed in the central nervous system. Are they expressed in neurons or glia, or astrocytes, etc.? When we do that, so this is 41 proteins that we handpicked that we knew that were only expressed in the central nervous system. Again, you might see some, if you squint, you might see that there might be some pockets of disorders clustering together, but clearly nothing really jumps out at you as being distinct patterns. So because we didn't see any clear, I'll say clustering again by genetic cause, we actually just lumped all of the affected samples together and we said, okay, because there's no clear reason for us to separate them by disorder, let's put all of our affected samples together and compare them to all of our control samples. So that's about 380 affected samples versus 150 control samples. And when you do that, if you look at this volcano plot on the top here, you can see that there's 160 upregulated proteins and 72 downregulated proteins. And we pulled out a couple here that just really popped out and have been implicated in other neurological disorders. SAA1, which is serum amyloid alpha 1 and 2, and then glial fibrillary acidic protein, the marker of astrocytic activation, was significantly upregulated in our cross disorder volcano plot. And if you look at the levels of these three proteins, when you are looking at all of the individual disorders, you can see that control average is right here on this top bar right here, and then the average expression value and the variability across all of the disorders. And then Kleefstra syndrome samples are highlighted right here in this pink bar. You can see that SAA1 is slightly upregulated, although for GFAP, that doesn't actually seem to be a remarkable increase in GFAP for Kleefstra syndrome in particular. Okay, so the next thing that we looked at was, okay, so there's no clear separation or similarities based upon genetic cause, but what if we started putting groups together based upon their shared molecular function? So this is actually really similar to how Dr. Harris was presenting, looking at, are there similarities in some of these biomarker profiles among chromatin modifiers? So we had a handful of other molecular groups: transcription factors, channelopathies, chromatin modifiers, copy number variants, synaptopathies, transporter kinase, and then one disorder of known as a demyelination disorder. So an example of what we were doing was we looked at copy number disorders associated with copy number variants. So we had Prader-Willi syndrome samples and DUP15 samples included in this project. The cool thing is that when you do this same UMAP, so just looking at a visual representation of how the samples are distributed, looking at all of the proteins, you can see that the samples that are associated with DUP15, which again, DUP15, as many of you know, is associated with a duplication of chromosome 15, cluster opposite from individuals who have Prader-Willi syndrome, which is a disorder associated with silencing of chromosome 15. So this was a really cool proof of concept to say, yes, these UMAPs are a good sort of general way of showing how things are distributing. So when we look at chromatin modifiers, so again, now we swapped out copy number variants and we're looking at samples from the disorders that are known chromatin modifiers. So these are samples from CHD2 related disorder, Kleefstra syndrome, Cohen-de-Vries syndrome, Kabuki syndrome, and then lastly, Tatten-Brown-Raman syndrome. And if you look just in general, almost all of the Kleefstra syndrome and Cohen-de-Vries syndrome and Kabuki syndrome samples are in sort of this upper right-hand part of the UMAP. So because of that, we thought, okay, well, what is it that might be causing all of these samples from these three different disorders to be similar compared to Kabuki syndrome and Tatten-Brown-Raman syndrome? So in order to say, okay, why are these clustering together? What we did was I took all of the Kleefstra syndrome samples and I compared the expression of those proteins compared to age-matched controls. And the same for the samples from Coulomb-Duffrey syndrome and the same for samples with CHD2 related disorder. So again, these are proteins that are significantly up regulated on the right-hand side and significantly down regulated on the left-hand side compared to control samples, unaffected age and sex matched controls. So you can see that there are quite a few down-regulated proteins, slightly fewer up-regulated proteins. But I started looking which proteins that were differentially expressed also show up or in common with Coulomb-Duffrey syndrome samples and CHD2 samples. And it's a little hard to see because the Coulomb-Duffrey's volcano plot, the scale of the x-axis is a little messed up. But I tried to highlight some of the two proteins that were in common between these two disorders. This is SHMT1, which I believe is an acetyltransferase, and then this CBLN1, which is actually, when I looked it up, was a synaptic plasticity marker, which is expressed almost exclusively in the cerebellum. So these are both two differentially expressed proteins that are shared amongst these two different chromatin modifying disorders. So these are examples of two proteins that we are now trying to see, do the expression of these proteins change with any of our outcome measures in the Vineland? So the other thing that we were looking at is really the issue of trying to figure out what is the best disease severity measure to sort of look to see if there are correlations with these protein values with that indicator of disease severity. And one of the ways that we were trying to figure out what would be the best phenotypic variable is to do this weighted co-expression gene analysis, which is you take all of the proteins, the 4,000 proteins on your platform, and you cluster them by shared biologic function. So the groupings here on the x-axis and the y-axis, the colors are completely arbitrary. But the important thing here, even though these heat maps are a little complex, is that you can see that these groups of proteins here, which correspond to just arbitrary colors on the y-axis, most closely correlate or most consistently correlate with age. You don't really see really significant impacts of epilepsy on proteins. Surprisingly, you don't see a giant impact on diet or collection time. And this is true when you look at all of the affected samples separately, when you look at things that correlate with proteins and phenotypic variables and the control samples as well, is age consistently seems to be the variable that impacts plasma protein the most. So because of that, we started looking at, okay, well, how many proteins significantly change with age in the data set of proteins that we're including on this platform? And we see 167 proteins significantly change in some direction, up or down, with age across controls and affected individuals. When you put these 167 proteins in a pathway analysis to say, what biologic function do these most have in common? The top molecular pathways are repeatedly related to immune function, cytokine signaling, neutrophil, or astrocytic activation, which really starts suggesting that some of the more inflammatory proteins or immune response pathways really change with age and might be in a disease-specific way. So I think this is one of my last slides here. So just to pull out what one of these actually looks like is here is a graph that just shows age and how S100A9, which is one of the pro-inflammatory proteins that came out from the 167 that changed with age. You can see that our affected samples are the ones in red circles, and the control samples are the ones in black or white circles. You can see that both increase expression fairly similarly with age. Sorry, let me go back one. Actually, the plot should have been replaced with what this looks like exactly, specifically for Kleefstra syndrome patients, and it looks almost exactly the same. So just pretend that this has been replaced with a figure that looks only at Kleefstra syndrome samples, and the change with age for the expression for this S100A9 looks exactly the same. Okay. So again, now we're looking to see, okay, does this particular protein, can it actually be a biomarker? Does it correlate with an indicator of disease severity that we collected as part of this project? And that, by and large, are the Vineland subdomains. So when you look at protein expression change over the composite score for the aberrant behavior, daily living skills, or communication, none of these correlations are actually significant when you look at these Vineland composite scores. Whereas the correlation was significant when you looked at age. There are a variety of explanations for this, which I won't go into right now, but by and large, that is what we were looking at right now. Okay, so current conclusions / next steps. So we found several proteins that are elevated or reduced across several disease groups, if not all of them. We're still working to figure out what really is the most appropriate phenotypic measure to really assess clinical severity, and likely this might be different for each disorder. We're currently collecting more cross-sectional samples for these same individuals to not only validate our initial findings related to these sort of pro-inflammatory and immune response protein hits that we've found, but we're also looking to collect longitudinal samples as well. All of our findings will be validated on this secondary proteomics platform called NuLISA. That data hasn't come in yet, but we're really looking forward to it. And then the other thing is that we're currently in the final weeks of collecting cerebrospinal fluid to start looking at how do some of these changes in plasma proteins change with age in neurotypical individuals. Okay, so here are the team of people who actually helped us collect all of these samples at roadshows. This was largely done by Sasha and Grace and Danielle and Taylor. We dress up as funny cartoons. Sometimes that makes kids cry more, actually. We had a lot of help with data collection from the Simon Searchlight and RareX, who actually helped us share Vineland data and Pels data. Matrix and Jason Colquitt, Andrea, are where we store all of our phenotypic data. My fantastic computational team, Ricardo, Megan, and Daryl. And then Natasha for helping us start figuring out how best to interpret our Vineland. This project was funded by these industry groups. They have been fantastic partners and they're interested in rare diseases. So thank you for their support. And then obviously, thank you to all of the foundations who recruited to make this project possible. Clearly, none of this would have been done unless individual PAGs like iDefine had actually recruited for this project. And I think that's it. I'm super happy to take questions. And just now I'm remembering, Eric, you told me to talk slow for the translators.
I think you did pretty well talking slow.
Okay.
I think that was good. Yeah.
I'm happy to take.
Now I have to talk slow.
Oh, okay.
That's going to be the hard thing. Fantastic. I can still remember Jeff already thanked everybody who contributed samples to this project, but I can remember when we were trying to get everyone to sign up and
2023. My gosh.
Yes, and rushing through that process, and it's so cool to see this start to come together. And I think the results really speak to the value of doing this kind of work across a lot of different disorders because of that ability to cross correlate and better understand what patterns are potentially meaningful what patterns maybe aren't. But yeah,
Mhm.
so interesting to see like KDVS show up with EHMT1.
Mhm.
So I
Yeah,
had a question about the fundamentals
sure.
of doing biomarkers in blood for disorders that are primarily disorders where most of the challenges come from the CNS. And I think I can answer my own question because it, it's looked like at one point you were looking at only at proteins that are expressed in the CNS, but you're looking at them in blood. So by definition, there has to be crosstalk between the blood and the CNS, or else those proteins aren't there to even be measured in the first place. Is that the answer as to why we think we can get at the CNS by using blood? Which is a fundamental question. I think it's, I guess,
Yeah,
being
and
asked by this study.
yep. So I don't know if this is the correct answer, but by and large, most of the let me go back to my slide that shows the groups. Oh, here we go. So by and large, most of the disorders, there's a couple exceptions, but for example, like Cancel 1 for Coulon de Vries is not explicitly only expressed in the brain. It's expressed in the periphery as well. And so that's one of the reasons why you could ultimately use a readout from something in the blood because it's expressed in the liver and it gets into the blood or the kidneys, etc. Now, does that mean that it could be doing something different in the liver than it's doing as a role in the neurons? Obviously, that could be the
Yeah.
case, but you know, that is one of the reasons why people have still continued to look at blood as a readout for something like that's happening in a CNS state. I will say though, it's pretty clear that looking at things in plasma is considerably noisier than looking at things that are from the cerebrospinal fluid.
Right. And it is obvious, given the neurofilament light results, that some things from the CNS absolutely do make it into blood and
Mhm.
will pass across the blood-brain barrier into blood.
Yeah.
But I thought you on one of your when you were doing your clustering slides, I thought there was one where you were looking only at things that were expressed in CNS.
Yep. So if
Yes,
you,
those 41 proteins are CNS only, and yet they are showing up in the blood. So they must be in some way crossing the barrier and getting out
yep, yeah,
to where they can be
yeah.
measured.
I
Yeah,
mean, neurofilament is only supposed to be expressed in neurons.
exactly. Yeah,
And GFAP that I know of, although somebody I'm sure on this call will be able to say that's totally wrong, I think is almost exclusively expressed in astrocytes. So like
yeah,
it is detectable, and the instruments and the technologies these days are so much more sensitive than when the dinosaurs in my lab were helping us run things. So yes, the short answer
yeah,
is yes, you can detect them.
yeah.
Um, whether they are great readouts for sort of the pathogenic consequences of EHMT1 in the brain, um, that's what we're still trying to figure out.
Yep. And I think this is biomarker discovery. This is not biomarker refinement. You guys are doing the early discovery work to start to
Mhm.
see if there's something there. Yeah. Other question I had was I know that there's intent with this project to not just look at protein, but also look at other markers. Do you have any comments on when that type of data is going to start to come in?
Wait, say that again.
Well, you're doing protein, but I think there was
Yep.
also going to be some methylomics, potentially
Oh, right.
metabolomics,
Yeah.
other
So
omics streams that will be reviewed using the same plasma inputs.
yeah.
Um,
Okay, so we have not figured out who we are going to collaborate with metabolomics on
okay.
metabolomics yet. That's still a goal. We were going to do it with Metabolon, but they've been backlogged and don't have the bandwidth to accept 2,000 samples just yet. However, they have said that when they are able to receive them, they still want to run this project. That being
Yeah.
said, I don't know when that type of data will be readily available, but we'll share it in the same way that we share the proteomics data. I think the vast majority of the samples that were included in this project are being run through an investigator at Emory, Victor Foundes, on the new LISA platform. which again is a proteomics readout, but it's a CNS specific readout and is supposed to be more sensitive for specific things. That again will be shared, I think, sometime in the next 3 to 6 months, but it will be shared in the same way that the SOMAScan data has been shared.
Great. And how can the community continue to participate in this? We did the initial first wave. I think you guys are collecting now for the second wave, right?
Yep.
Because you want longitudinal.
Yep. Yeah. I mean, so the easiest way is to go to these things we call road shows. Let's see, what's today? The end of September. So the next one that we're having is at the beginning of December in Atlanta. So if you have anybody who lives in Atlanta or can get to Atlanta, there's some paperwork involved in terms of providing consent and giving us your genetic report. But other than that, it's an issue of walking in and donating and leaving. I will say.
And are you taking, since you're doing longitudinal now, are you taking also new patient samples who weren't in the first cycle? So you can, so if you weren't part of the first cycle donations, you can absolutely still go to one of the road shows.
Yes, definitely.
Okay.
Yes.
Yeah.
Yeah.
So yeah, speaking for ourselves, we will definitely recirculate that schedule with the community.
Perfect.
And distribute the information on how to participate.
Yep. Great. Fantastic. I will say one thing that I have done a less than great job at emphasizing is, we have some proteins that seem like they could be candidate biomarkers. We really need to either push the collection of everybody's favorite surveys to complete, to sort of start looking at these protein levels change with some indicator of clinical severity, whether that's the Vineland or whether that's the ORCA or whatever assessment that the Pfalzer community thinks would be the best. That is like one of the biggest challenges that I underappreciated at the beginning. At the beginning, I was just worried about getting patient samples. And now I realize I don't have phenotypic data to compare it to.
So if you've participated in the biomarker collection, you have to make sure to go in and do your surveys.
Yes.
That's the message. That's the other thing the community can do. So
everybody's favorite stuff.
Yes. And if you're not sure how to do that, reach out to Jeff and to me if you're in the KS community, and we will get you set up. Okay. I don't see any other questions. Jeff, did you have any questions or comments? So you just jumped in.
Yeah, just jumping in. I mean, I think that we've heard a lot about surveys today, and I think that there's an opportunity here to piecemeal this out because this information is so critical, it's so important. But then there's the reality of just time and bandwidth and energy to be able to fill them out. And so we'll do something where we're just kind of, again, piecemealing it out, where maybe it's Sunday surveys that we promote certain surveys on social media every single Sunday, and then that way it's digestible. It's one breadcrumb is trying to eat the whole loaf kind of thing. And so but it's, again, this is where the community and an engaged community has immense value to all of our partners, is our ability to contribute and add value to these studies that frankly they just can't get anywhere else. And so I don't think we can underestimate the importance or overemphasize it. And so but it's incredible work, and it's exciting, and I'm excited for round 2 for the road shows. And again, to the point of, we do have the opportunity for mobile phlebotomists, so don't let that be a detractor if you can't get to the road shows. The road shows are just already built and super efficient for everybody, but that's not to say that our goal is to take that number of 11 and at least double it, of the 0 to 17 range. Our community knows that we look at these things in a fun, competitive way from time to time. And so when I see other organizations that are ahead of us, it's we have an active community. I mean, you can see it with our map, and we need to see that across all of our efforts. So that's it.
I will say one last thing is that we have tried very deliberately to reduce the amount of survey burden that we impose on caregivers and family members as much as possible. And what I mean by that is not just hand waving, is we have data sharing agreements with Simons and RareX for most of the surveys and assessments. And so and it's bidirectional. So if you complete a series of surveys on the Combined Brain platform, you yourself, you meaning the caregiver, you can download that data and send it to whoever else. Like if you're participating in a survey or study with Dr. Harris, you can send your data directly to her and say, you know, I actually don't need to take the Vineland again. I just did it for Anna at Combined Brain. So good God, here, there you go. But we can also push it. So it's always important to, before you do things, say like, I just did this on that platform, because we are slowly but surely trying to figure out a way to talk with each other and share. And for this project, we pulled in Vinelands from Simons and RareX so that we didn't have to make Eric fill out the Vineland again, although he probably wouldn't have anyway. But that's the idea. So yeah, we know
Filled it out.
we know that it's a pain, and we are all slowly but surely trying to figure out a way to collaborate with each other so you don't have to do it 4 times in a year. And you're not supposed to do that anyway.
Yeah. So Anna, what would be super cool for the community would be is if there's some kind of a how-to guide that you guys come up with that we can just distribute that says like how to get your stuff reused so you don't have to get survey fatigue redoing things over and over.
I can tell you the easiest way to do that is that Simons and RareX have a place for the CRID. So if you have your CRID on your account on those platforms, that's what we do. We say like, I need Vinelands for all of these CRIDs. I need Vinelands for all of these CRIDs. That's the only thing. Don't do any more work other than that.
Okay.
Cool.
Cool. Yeah, we'll have to do some discussion with the community on refreshing on CRID. But if you have done a sample donation to the biorepository, you have a CRID. You have to have a CRID. So in that case, you have a CRID. So if you're looking to fill out surveys, but then who do they tell, hey, I don't want to do the Vineland twice, I already did it here, to get the data moved around? Or is it just sort of automatically going to happen if their CRID is in both?
For us on our consent form, it has a box that says, are you on Simons? And it clicks the box. And so we need to reach out to the Simons team and say like, here's a CRID, do you have it for them or not? It obviously doesn't always work that way. So some of it probably lays on the, the caregiver to be like, I swear to God, I just did this. And
okay. Well, let's work on that offline. I think it'd be cool if we put some little guide together that says like, look for this, check this, totally. and then it's super simple, and we can use that. All the people in Combined Brain can use that and make it more efficient for all the groups.
Yeah.
Because survey fatigue is real for sure.
Yep. I hear you.
Yeah. Okay. Fantastic. I don't see any more questions. Thanks so much, Anna.
You're welcome.
Yeah, and I just want to reiterate for the community, this project is impossible for us to do on our own. Impossible. This is a project you have to do with a large group of aligned people, like we're doing with Combined Brain. So it's an and the companies that are contributing it. So it's an amazing project, and it's truly unique what's being done here. So amazingly exciting.